After the gene replication and the production of the new gene, the template after the template and then the production of the new gene, now is the process of the gene packaging. How the new material has been packed inside the capsid. For this purpose, the capsid of the virus plays very important role, and we will see this in detail. The gene packaging is the fundamental step in the virus life cycle because after the genetic material and it has to produce or replicate the new genome. So after the production of the new gene, how it has to replicate, how it has to packed. Usually it is performed through the capsids because the capsid's length depends on the gene's length. If the gene of the virus is bigger, the capsid will also becomes the big. And similarly, if the size of the viral gene is smaller in the viral genome, we can say, similarly, the capsid will also be the smaller. So the packaging of the virus, the packaging of the genome of the new virus mainly depends on the capsid. Capsids are also reformed sometimes for this purpose because it has to surround along with the genome. If the new genome is produced, the capsids, it is the because the structure become the protein. So the host has already have a lot of proteins surround inside. So there are the capsids. They are also sometimes reformed and they surround the genes. Similarly, the packaging of the motor proteins which are produced by the hydrolysis of the ATP inside the body after the genetic material. So there are packaging of the motor protein and condensed genome into the confined place. Both of these functions are usually happens with the help of the capsid because capsid has to condense the genome into one confined space, just like you can see in the picture. Whole the genome is just happening inside the body of the host, so it has to package, it has to condense in one place inside the body, and then the motor proteins, they are always there, and they have to be condensed also so that they can be packaged with the virus. There are different steps involved in this process. We will see these steps one by one in the coming slides. Binding of the terminase. First of all, the step 1 is the binding of the terminase, and then step 2, binding of the pro-capsid portal, then DNA translocation, and then contamer cleavage. These are 4 different processes which are involved in the genome packaging, and we will see them one by one. The step 1, the terminase binds to viral genome. Just like you can see there in the pictures in the diagram just in front of you, the terminase, the first step, the terminase binds to the viral genome. This is, you can see the yellow color terminase, and this is the new viral genome which has been just produced. It may be the RNA or DNA which has been shown in front of you. Viral genome concatemer, and then the terminase, it will be just binds to terminase so that all the process can be terminated. Terminase DNA binds to pro-capsid portal. The step 2, the pre-assembled capsid or pro-capsid, usually they will just bind the terminase, and after it is terminated, it will just bind with the pro-capsid, and the terminase DNA complex binds to the portal of the entry on the capsid. Just clear from the diagram given here. Then the DNA translocation. DNA translocation happens with the portal structure provides the entry to the DNA inside the capsid, and then packaging the motor proteins after the termination. Now the large terminase comes into action, and it will be just like the clear from the picture shown. It will be just the DNA translocation inside the capsid. The fourth process or the last process is the contamer cleavage. Because after the packaging of the DNA, the remaining DNA has to be terminated, has to be wetted. So for this purpose, just is clear from the diagram it's given below, the DNA cleavage and completion of all the genome packages occurs with this process.